Right, let's get started, guys. We will be finishing up lecture three, 15 minutes, 20 minutes, and then we're gonna start on number four, okay? And then I'll tell you more about the quiz that is going to be on Tuesday. I know you guys had a chemistry quiz this morning, right? Yeah, but try to summon your strength, okay? And we're gonna be done in 2 hours. Here we go. The next few slides is going to show you organelles that are not found in animals. Some of these are found in plant cells, and some of these are going to be found in things like amoeba, paramecium. We call those things protists, right? So the first thing is chloroplast. Chloroplast are these organelles that enables plants to do photosynthesis. And photosynthesis is basically a reaction that allows plants to use the energy from sunlight and make their own food. Okay? We have to consume food in order to make energy, but plants, they can make their own food. Right? And chloroplast is what allows them to do it. So the chloroplasts are found in plant cells and some protists as well. Okay, protists are these they're not animal, they're not plant, they are like those amoeba that I've shown you, right? The paramecium that I'm showing you. Okay. So some of them are also photosynthetic. The chloroplast contains a compound called chlorophyll, and that is what enables them to capture sunlight. So chlorophyll makes the plant green. Anything that's green, like the leaves, right? Even part of the stem, they contain the chlorophyll. Just like the mitochondria that we did last class, chloroplasts are also double layered membrane. It has an outer membrane and an inner membrane. You do not need to memorize the structure of chloroplasts. Okay, we're not going to be focusing on plants in the course, so no need to memorize. The relationship between chloroplasts and mitochondria is an important one. Okay, the mitochondria is, as you learned last class, the location for energy production. And so how do you produce energy? You take glucose and you react it with oxygen. And then in the mitochondria, it will convert it into carbon dioxide and water. Okay. So our cell use this mechanism to produce energy. For plants, the photosynthesis process occurs in the chloroplast, and they need to take carbon dioxide from the air and water from the roots. With the help of sunlight, it gets converted into glucose and oxygen. So in other words, the ingredients for the mitochondria are actually the products of chloroplast. And the products of the mitochondria are the reactants for the chloroplast. So they complement each other. Do you guys see the relationship? The things that the mitochondria makes is going to be used by the chloroplast, and then they make the things that will be used by mitochondria. So which one do we have, guys, in our cells? Mitochondria or chloroplast? Okay. Which one do plants have? Both of them. Both of them. Right? So the difference is we have to eat the food and then we burn it. Plants, they make their own food with the chloroplast and then they burn the food for energy. Okay? The sunlight doesn't give them energy. The sunlight gives them food. And then their mitochondria will burn the food and make energy from that. Okay? Here is another structure that is found in plant cells. It is called the central vacuole. It's really big. You can't miss it. If you have the labeling sheet, you can take a look at it. On the back is the plant one, and you can see there's a big structure, right? At the bottom left there, that would be the central vacuole. Okay. So the central vacuole is primarily for storing water, but it can store other things like nutrients and even the colors for the petals. And in some cases, if the plant is poisonous, they will store the poison in the central vacuole as well. It's a really, really big thing. You can't miss it. Okay. Animal cells do not have a central vacuole. There are other kinds of vacuole. Okay. Right here, we have this guy again, I showed you earlier. The name is Paramecium. You don't have to know how to spell it. I'm not going to ask you to, talk about this thing. But they live in fresh water. And one of the challenges of living in fresh water is that the water will keep on coming into the cell. Okay? And if they don't pump out the excess water, then it's going to swell up and eventually explode the cell. It will burst and die. Right? So they have these special contractile vacuole, which are basically pumps that will send excess water out of the cell. And it prevents it from swelling up too much and dying from it, basically. Okay, so these are, you have a contractile right here, contractile vacuole here, and then here. Oh, it's already labeled for you, okay? So basically, it's going to pump water out of the cell. Okay? We do not have these things, okay? It's only found in some of these protists. There's another structure that plant cells have that we don't have. They are called cell wall. It's not the first time we learn about the cell wall. You should know from lecture 2 that cell wall are made up of cellulose, which is a type of polysaccharide. It's very strong. It protects the plant cells from bursting. Okay, you got to water your plants frequently, and all those water is going to be absorbed by the cells. It's going to be stored in the central vacuole, but that would mean the cell is going to swell up and get really big. You don't want it to burst, so they have a cell wall to protect them from it. Okay. All right. Let's see how much you remember from last class. We are looking for all the false statements. I got to read it first. All right, I got it. Let me know what you think. First 3 are false? Yeah, okay, maybe. Anyone else? Okay, just show me your fingers. Still in chemistry mode, huh? No space for bio. That's all right. Let's take it up. We're looking for the false statements, the one that are incorrect. So let's see. Smooth endoplasmic reticulum is part of the endomembrane system. That's correct. Okay. The endomembrane system includes the following. Nothing ever works. All right, so the endomembrane system includes the following, okay? Nuclear envelope, rough ER, the channels with the ribosomes on them, right? Smooth ER. Right Golgi body, and one more. Lysosome. Okay, so these are all part of the endomembrane system, and as you can see, smooth ER is part of the endomembrane system, so that's correct, and we're looking for the false statement, okay? Number 2, there is an abundance. Abundance means a lot of mitochondria in muscle cells. Is that true or not true? It's true, right? Mitochondria, they are powerhouses. They make energy. Muscle, of course, requires a lot of energy. So that's true. Next one. All cells have cell wall to protect them from the environment. Yes? No? Do we have cell walls, guy? No, we don't, okay? No, we don't, okay? So this is false. Animal cells. Animal cells don't have cell wall. Okay, look at the diagram, guys. We don't have a cell wall. We look at the plant one at the back. There's like an extra layer, right? That's the cell wall out there. Okay. That's why animal cells, the shape are less rigid, but plant cells are very rigid. Hydrogen peroxides are found in lysosome. That is false. Hydrogen peroxides are found in peroxisome. Right? Peroxisome. These are dangerous reactions that happens, and we need to contain them, and it all happens in peroxisome. Lastly, chlorophylls are found in chloroplast. Yes or no? Yeah, true, right? We did that 5 minutes ago. Okay, so this is how you should do the questions on quizzes. All right. I look at your quizzes. There's not a lot of markings on questions. I mean, some of them you just kind of know, but for more difficult ones, you need to be writing over the choices, right? You need to be choosing it for good reasons. Okay, it's not like, oh, I feel like this is the right answer, let's do it, right? Okay. So make sure you mark it up. It's a good strategy for doing multiple choice questions. Okay, I have good news for you. We are not gonna do this. You don't have to study it. 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 No. Okay. Got it? I'm serious. Okay. You guys look shocked. Okay, so 1, 2, 3, 4, 5. Okay. I'm not gonna test you on it. Okay. If you really want to learn it, come see me, after class or something. I give you a personal lesson, okay? But otherwise, don't worry about it. We don't have enough time. We will do the centrioles though. Okay, centrioles. Centrioles is another organelles. They are found in animals, but not plant cells. Their job is to produce a special kinds of fibers. They are called spindle fibers. We'll learn more about them later in the course. For now, it's enough to know these spindle fibers will help guide the movements of chromosomes. Remember I told you when a cell divides, you need to make sure each cell gets the same number of chromosomes, right? You get 46, you get 46. So something has to guide the movement of these chromosomes, and that will be the function of the spindle fibers. Okay, can you find the centrioles in the picture here? Can you find the centrioles? Where is it? Give me a rough direction. Top, bottom, left, right, center. Where are the centrioles? You could, or you could look for it. Right here, right? Do you see it, guys? That's a centriole, okay? It looks like pasta that has been uncooked, okay? There you go, 2 of them. All right, so they produce spindle fibers. All right? Listen, I cannot stress the importance of this diagram. You gotta do this because on your quiz, guarantee you will see either this or this, depending on which version of the quiz you get, okay? And it's going to be 5 labels. I'm gonna point to 5 things and you have to label it for me, okay? Now, I don't have time to go through this with you in class, but you have to do it. And there is also, I'm gonna show you once again, in case you are having trouble, and we have some newcomers in the class as well. Go to the course page, and there is quiz number 2, which again is on Tuesday, okay? Don't miss it. There is the review video for it, right? So watch it And at the end, you see, I went through labeling the cell with you. Do you understand? Okay, so just do it yourself first and then go watch this thing, okay? And you should watch the whole thing, of course, just like last time. I think I told you already, but let's go through it again, whatever. It's gonna be proteins and nucleic acid from lecture 2 and then all of lecture 3. For lecture 3, I cross out some of the stuff, right? You're not responsible for them, obviously. Okay. 15 multiple choice questions like last time, 4 short answer questions, and 5 labelings. Okay, 5 labelings. No short form. Don't tell me RER, okay? I will circle it and put question marks and not give you any marks, okay? Rough endoplasmic reticulum, smooth endoplasmic reticulum, okay? No, yeah, that would be too easy, okay? So no labels, okay? You have to know how to spell them, okay? You will just get the diagram with the arrows. Is that clear? That's 5 marks out of I think 30, okay? That's a lot of percent. So please make sure memorize this. Okay, I have 2 more for you. We have cilia and flagella. Cilia usually covers the entire cell, and it's made up of the same stuff that makes up the spindle fibers. Okay, so the paramecium I showed you, with the contractile vacuole a few slides ago, they have cilia that covers them, and it helps them swim around basically. Okay, so these are cilia, little hair that cover the entire cell. We also have cilia inside our body. Right? We have cilia in our airway to help sweep up a mucus and trapped dust particle. Right? It goes up to your throat. You can spit it out. You can swallow it. Right? That's in the airway. For female, they also have cilia in their fallopian tube. That's the tube that connects the ovary to the uterus. It helps guide the movement of the eggs to the right place. Flagellum. Okay, flagellum is a tail. It usually a cell will only have 1 or 2, and basically cilia goes like this, back and forth. Right. A flagellum would be like wave-like, whip-like motion. So for humans, the only place you will find flagellum would be the sperm cell, and they only have 1 tail. Okay. If it has 2 tails, then it's not going to be able to swim forward properly. But some organisms, they have 2 flagellum. It's like those protists I tell you about, right? They could have 2 tails, and it's fine for them. Okay. So both of these things are made up of something called the microtubule. It's the same stuff that makes up the spindle fibers. That's all you have to know. This is the airway. You can see it's very nice cilia. For smokers, they will damage the cilia. They will no longer be able to sweep up the trap particle. So long-term smoker develops what's called like a smoker's cough, right? They have to constantly, cough the trap particle out and then they spit it out. This is the fallopian tube I was telling you about. I'll spell it out for you. Fallopian tube or oviduct, same thing. And that's where the egg is going to be. And then these little cilia, the hair, will help guide the egg towards the uterus. Okay. And that's all. This is just summary of some of the characteristics of animal cells and plant cells. I'm sure you can read that on your own. Don't worry about this. We didn't do it. And that's it. Okay, that's the end of quiz 2. Do we have any specific questions? No? Yeah, that's okay. So next week is a busy week for you guys. Because I made a schedule, yes. Let me see what's happening here. Yeah, in my class, yeah.
That was, I didn't know that.
Oh,
We have your quiz, the chem quiz, and math testing.
yes. Very busy. Hold on, I don't know what I'm looking for anymore. Oh, there it is. Okay, this is not your class. Is this it? This is it. Okay. So Tuesday is the quiz, like I said. Okay, be on time. It's gonna be in the other room, but same thing. I know you guys like to hang in the room before I come. Okay, but next Tuesday, just wait outside. Okay, buy yourself a coffee or donut or whatever. Okay, stay outside because I gotta set up the room, right? I gotta put the papers and everything. So please give me some time to do that before class starts. Next Thursday is your Beetroot lab. You have to be in class in order to do it. I'm gonna let you guys make your own group later on during the break. You can write down group one and who you want to be in that group. Right? I think most of us are here today. I feel like it's a pretty full class. 3 to 4 people per group. Okay. If you are not in a group, when I take this home today and you're not in a group, I will force you to be in a group. Okay. It's much better. You choose your own people. Okay. You choose your own partner. Okay. Do you understand that? Everybody in the group is going to get the same grade. All right. I just want to put that out as a disclaimer. I don't want to deal with situations where you come to me after and say, oh, so and so didn't do any work. We did all the work. Okay. They should get 0. I don't care. Okay. You guys sort it out. Divide the work equally. You only have to submit 1 lab report per group. Do you understand? All right. Now, to prevent problems from happening during the lab, I want it to be a smooth, seamless 2-hour lab session. Please read the lab this weekend before you do the lab next week, okay? If you don't understand what's going on in the lab, you will see me on Tuesday and I can, answer your questions. But you gotta read through the procedures. It's not that complicated, okay? And you need to maybe write down a flow chart perhaps. So when you arrive, you know exactly what needs to be done. Okay? I'll try to explain a little bit more on Tuesday if I have time. But that's what it is. I will leave it on the side.
You want to write flow chart before?
Sure, yeah, but you don't have to hand in the flow chart. It's not for about, okay? I'll leave it here. You pick it up during the break. This is the lab report. This is the lab report. The lab report, again, 1 copy per group. And you are not handing in physical copy, okay? This is just for you to write down things during the lab because there will be data you need to collect during the lab. So you can do your rough work here, and then you will have to type it up and then submit it. You will have to type it up, and then you will have to print it out, and then you have to submit it to me. Okay, so you cannot submit this copy because you got to do graph and stuff like that, and you have to put the graph in the report. So each group will hand me a physical good copy. Does that make sense?
Yeah.
Okay, so everybody will get the same mark on the report. All right, so pick this up as well when you are going on your break. And I will have the sign-up sheet here for you as well. Okay, just say group 1 and then write down the names and then put a bubble around it. All right, if you go by a different name than the one that is on the class list, please put that in bracket so I can find you. Do not wear fancy clothes for Thursday. Right? The beet juice is very difficult to get off. I got a person who wear all white to the beetroot lab. And I offered her an apron, but she didn't want it, okay? And guess what? The juice got on her white clothes, right? So don't wear anything fancy, wear dark colors. If you have aprons, you can bring your own, but we have some here. We have aprons here. But if you don't want to use other people's apron, bring your own. You don't need safety goggles. You don't need gloves or anything like that. It's nothing dangerous. It's beetroot, okay? You eat that for salad, right? So that's that. 10% of your grades. Yeah, easy 10% if you do the work right. We good? Excellent. I'll tell you more later if I have time on Tuesday. Okay, today we are gonna be doing lecture 4. Lecture 4 is not on your quiz, and it's about the cell membrane. So we're gonna start by my favorite activity, drawing. Let's do this. Yes. Yeah, we're drawing. So here is your paper sideways. Okay, we're just gonna be drawing something like this big. Okay, and then we'll have some writings here, some writings here. That's it. Okay, it's gonna take 10 minutes. Right, and the title of the note is Fluid Mosaic. Model of the cell membrane. Is it going to help if I show you the final product so you can plan ahead?
Yeah.
Yeah. There, this is what we're drawing, okay? Right there.
Yeah, that's not terrible.
Yeah,
It's better than last.
sure. Fluid mosaic model of the cell membrane. So our cell membrane, follow this model. But what does it mean, right? And first of all, it's fluid, meaning it is not solid like a wall. If it's solid like a wall, then nothing goes through, right? Remember, the cell membrane is selectively permeable. It needs to help regulate what goes into the cell, what leaves the cell. Right? You want nutrients to come in, harmful stuff to stay out. So, you cannot have a cell membrane that is completely solid and rigid. And for that reason, it is going to be in a fluid state, right? It's dynamic, it could change, right? It's not fixed in place. Number two is mosaic. I think I'll just show you what a mosaic window is in case you don't know. Mosaic window. It's one of these things, right? Okay, so the whole window is not just like these regular window. It's made up of different pieces, right? All connected together, and it forms the whole window. And that's what the cell membrane is like. It's not uniform. It's not the same everywhere. Have you guys learned the word homogeneous in chemistry yet? Yeah, it's not homogeneous, okay? It's not the same thing throughout. It's more like a pizza, if you would, right? There are different components to it, right? That's why it's called mosaic. So here we go. We have our cell and the nucleus, and we're going to take a slice of the cell membrane, and we are going to take it out. Now, by now, you should know the cell membrane is made up of phospholipids. So I'm going to draw some phospholipids here. And the phospholipid is special because it has a hydrophilic head and two hydrophobic tails. Some tails will be saturated fatty acids. Others might be unsaturated. This is old information, right? So, you can put in unsaturated as you see fit. So that's one layer of it. And then I'm going to draw another layer below it. So it's like a sandwich, okay? Now I'm going to ask you to leave a gap here. And then just draw this one more time. And then leave another gap. And then draw it one more time, okay? It doesn't have to be same size. I just want you to make sure you have two gaps here. Okay, I'll tell you what the gaps are for later. But this whole thing is called the phospholipid bilayer. There are two layers, outer layer and inner layer. And this whole thing is going to be your cell membrane. Now, I want you to also understand that we are just looking at a section of it, a cross section of the cell membrane. The actual cell membrane is like a sheet, right? Now, don't draw this, but take a look first. The whole thing is continuous, right? So it's gonna be 3D, and then you would see more hydrophilic head. Do you understand? Yeah, it's like think of like a mattress, right? Okay, it's the whole thing. But we take a big knife and then we chop it, and we're just looking at a section of it. Do you follow? Yeah. So to make it not as complicated, I'm gonna not make it 3D. Okay. All right, I'm gonna give you a moment to catch up. So, just to help you recall some information, this is a phospholipid and it has the hydrophilic head. And then these are the hydrophobic tails. And like I said, the tails, they could both be saturated or it could be one saturated, one unsaturated. Okay, it could even be both unsaturated. So I'm gonna make a quick note here. The phospholipid molecules can move freely within its own layer, but they rarely flip to the other layer. Yep. Hold on, I will show you back. Put that back up in a second. So do you guys know like ball pits, like these things, right?
Yeah.
That the kids love, right? Okay. Think of the phospholipid bilayer like a ball pit. Okay. These little heads, okay, like the ball pit. The balls can move past each other, right? I can go front, back, sideways, whatever, right? So that's what I'm saying. These phospholipids, they can move around, they can bop around like those balls in the ball pits, okay? But they don't flip to the other side. They're gonna stay within their own layer. This one, sure, it can go sideways and go into the screen, out of the screen, but again, it doesn't flip. So no flip-flopping, but moving within its layer, it's okay. Right? And that's why this is called a fluid mosaic model, right? The keyword being fluid, as in they have the mobility to move around. If it's solid, then it's fixed in place. Like if you look at a brick wall, the brick's not gonna be like shifting from one place to another, right? But this is not solid. This is fluid. Is that okay? Yeah. Question? If you have questions, just ask. Okay? I'm sure other people have the same question. There are no bad questions. All right. Phospholipids are the major component of the cell membrane, but it's not the only component of the cell membrane. In fact, phospholipids, they are not very good for maintaining integrity. If the whole thing is made out of phospholipids, then it falls apart very easily. So you do need to have some kind of bigger molecule to help anchor everything in place. And that's where proteins are going to come into play. So in these gaps that you have created, you can put a protein in there. Okay, keep this in mind when you're doing the lab next week. Okay, so we have proteins that are going through the entire membrane, and we have proteins that are just going to be like attaching on one side of the membrane. If it's attached on one side, either outside or inside, we call it periphery. Peripheral protein. The definition is in the PowerPoint slides, but you can write it if you want, okay? Peripheral means either outside or inside. Whereas these ones, that goes all the way through, okay? These are called integral protein. Do you understand the difference? Okay? It's just going through or not. Okay? Think of this as a sandwich, right? Things goes all the way through or not. Yeah, peripheral means side, right? Like think about like your peripheral vision, your side vision, right? The peripheral protein just means it's attached on outside or inside. Okay, it doesn't have to be on the edge necessarily, but like it's the point.
So from one side it's connected, from another side it's not.
It's not connected to the other side. No. If it's connected to the other side, then it would be the integral protein. Okay, think of the pizza, right? Pizza, if you have the olive on it, The olive is just on the surface, right? It doesn't go all the way through your pizza, right? Like if you look at it from the bottom, you're not going to see the olive, right? Okay. That would be like the erythroprotein. You understand that? Okay. Integral protein, I don't know, like, sandwich, and then you poke carrot stick through it or something. Okay. That would be integral. Everything is better when you think about food. Okay, so these proteins, they have a lot of functions, okay? In fact, we're going to learn about 6 functions related to these proteins coming up. For now, we're just going to leave it the way it is. Now, there is another kind of lipid that will be part of your cell membrane. You did that on quiz 1. Does anybody remember what it is? Another lipid that is found in the cell membrane. I hear a lot of whisper. What is it again? Okay, we have phospholipids already, but there is another kind of lipid that will be in the cell membrane. Steroids, that's right. Specifically, which kind? Okay, yeah. Cholesterol, okay? Cholesterol. So there is the cholesterol. So these are cholesterol. Not a lot, just a bit. Okay, and the function of the cholesterol is to help control membrane. You know what? Let's say regulate. That sounds better. Regulate membrane fluidity. Now, remember, saturated fatty acid, they tend to pack together, right? If things pack together easily, then is it more solid or is it more liquid? That's right. If things pack together easily, then it becomes more solid, right? Okay. So the cholesterol is there to keep the phospholipids from coming too close together. Okay. It prevents this one and this one from like sticking together, right? So in other words, the more cholesterol you have, is it more liquid or more solid for your cell membrane? Let's think about that. Okay, if you think it's more, I'll repeat the question. Okay. If you put more cholesterol in the membrane, do you think is it 1 more fluid or 2 more solid? Show me what you think. More fluid or more solid? If you put more cholesterol in. Okay, anyone else? Okay. More fluid or more solid with more cholesterol? Change your mind, huh? Okay, good. Pick something, guys. It's okay to be not correct. Good. Okay. Anyone else? Yeah. So remember, cholesterol keeps these things from sticking together, right? If they don't stick together, then it's going to be more liquid. So by having more cholesterol, it will be more fluid. Do you understand that? Okay. Think about it. When things become solid, when things freeze up, they clump together, right? Like things stick together. So if you don't let them stick together, if you keep them apart, then it's going to be more fluid. If you have less cholesterol, then it's going to be more solid. So depending on the kind of cell membrane we are looking at, the cholesterol content would vary from one cell type to another. And that is an important point to remember when you're doing your lab report. Okay? What was the point? Depending on the kind of cell that we're looking at, the cholesterol content might change. Okay? More on that in a bit. All right, couple other things we're going to add to this picture. Over here, some proteins, turns out, they would have sugar chains attached to them. Okay. So this is carbohydrates. carbohydrate. And sometimes lipids would also have carbohydrates attached onto them. Okay, these are little sugar molecules. And then you're gonna have what's called a glycoprotein. Okay, glycoprotein. I'm gonna put it over here just so I have space. And this here is called a glycolipid.
It's about one on the
Mhm.
middle. Is it like?
Yeah, so if it's a protein with sugar, then the whole thing is called a glycoprotein. A glyco means sugar, a
Yeah.
sugary protein. Okay. If it has lipid with some sugar, then we say glycolipid. I'll tell you more about these things later, but they play a role in cell identity. Okay. How does your body know what belongs to yourself and what comes from outside? Right, let's say a bacterium enters your body. How does your cell know that does not belong to the body? Right? So part of the explanation is that they will check the kind of glycoproteins and glycolipids the cell has. And all your cells will have a unique set of identifiers, right? That are unique to yourself. No one's gonna share the same thing as you unless you have a what? That's right. Unless you have an identical twin, no one is going to share the exact same cell identity as you, right? Which is why, like, if we're looking for like a transplant, you can't just take a kidney from someone and then put it into someone else, right? Okay. The body's going to reject it. They're going to attack it because the cell identity are different, right? So you cannot find perfect match. Right? You try to match as much as you can, and then you actually have to put the person on immunosuppressant to dampen, to lower the immune system so that they don't reject the organ, right? Okay. So that's cell identity. And part of it is because of this glycoprotein, glycolipids. Is that okay? Mhm.
For the glycoprotein.
Yeah.
Never mind.
Okay. You can ask again later if it comes back. Okay, we're just gonna add one more thing, guys, to this picture, and that is these little things, okay? Now, I know I skipped all those slides, and I said it's not gonna be on, but these things are cytoskeleton. Cytoskeleton. Just to keep it very simple, cytoskeleton are basically they are like proteins that are found throughout the cell, and it provides support to maintain the shape of cells, for example. Right? So over here, all these cytoskeleton, they form scaffold for the cell membrane. So the cell membrane is not going to collapse on its own. Okay. You know how when people build new buildings, right? They need to have scaffold on the outside, right? Until the structural integrity is sound. That's what these are. So as you can see, the cell membrane is made up of many different components, all of which contributes to the function of the cell membrane as well as the structural integrity of the cell membrane. And that's why it's a mosaic model. It's not one thing, it's many things. Okay? I will not ask you to draw this out on the test. Right? Some people get very concerned. In fact, there will be no drawing. The quiz will be no drawing at all. Okay. I'm only drawing it out because it helps you guys understand better. At least I hope it does. Okay. Let's see, lecture 4. Okay, so we did that already. There is a question here. You can read it on your own later, but I'll just summarize the answer here for you, okay? Let's do a chart, not a very big one. Don't worry. Okay. Okay, so let's say we have environment here. And we have adaptation. Percent saturated phospholipid. Percent of unsaturated phospholipid. Okay, I'll draw a picture to help you visually distinguish the two. And percent cholesterol. So let's say we look at a plant that's growing in hot environment, okay? Like the desert or something, like a hot environment. What kind of adaptation, what kind of changes, modification the cell membrane needs to have in order to withstand the hot environment? Okay. So I want you to talk maybe talk with each other first, and then we're gonna get, is that a question or the answer? Answer, okay, maybe in a bit, yeah. So talk about with people around you, right? Think about, if it's in a hot environment, do you think the cell membrane needs more or less of this stuff, right? Do you understand? Yeah. So I'm looking for like higher, lower, higher, lower, higher, lower, right? You choose one. And then you gotta describe the adaptation. The adaptation, the answer is, do you want it more solid or more liquid for a hot environment? Okay? You understand? We're talking about cell membrane of a cell from a plant that is living in a hot environment. Do you want the cell membrane to be more solid or less solid? And if after you choose that, in order to have that adaptation, do you want more of this or less of this? Okay? No, 2 minutes. Go, go, go. The end. I have your quiz. Come on.
Cells located all over your body.
All over the body, yeah. So like if I scrape some thing off my screen, skin, put it under a microscope, you can see it. Body. Christine, are you here today? Christine, no, Christine. Mariana, are you here today? I don't think so, right? No. Mariana, no. Already. So if it's in a hot environment, do you want it to be more solid or more liquid? More solid or more liquid? Solid? Any group thinking more liquid is the right answer? No? Yeah? Okay. The correct answer is more solid. Why? Here's why. If it's hot, then it's gonna melt, right? Okay. You don't want it to melt. How do you make it not melt? You make it more solid. Okay. Think about it. The pan is hot, right? If you put butter on it, then it turns into a liquid, right? Right? So like you want to keep it as a solid form. You don't want it to turn into liquid easily. You make it more solid. To make it more solid, do you want more saturated fat or less saturated fat? More. And then this one, less. And cholesterol? Also less, because I told you less cholesterol is more solid, right? Okay, everything is in the notes. Okay, then cold is the opposite, right? Hey, try to understand it, okay? Don't memorize it. All right. I have people who just memorize, okay, more, less, 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 more, more. And then I switch the column on the test, and then it's wrong, right? Okay. So try to understand. You don't get this? No? Okay, do you have a question though? Okay, so your question is why do you need it to be more solid in a hot environment?
Yeah, also like yeah.
Sure. Okay, so one more time for those of you who might not understand this the first time. If the environment is hot, do you agree it is more likely to melt? Right? Because it's high temperature, things are going to turn into liquid, right? So that's why you want it to be the opposite. If you don't make it the opposite, then the cell is just going to melt because of the high temperature, right? So to make it not melt, you want it to be more solid. You want it to have a higher boiling point, in other words.
But this is not what actually happens.
No, this is
what-
actually what happens. Yeah. So
Oh.
if you look at the cell membrane of a cell that's taken from a hot environment,
Yeah.
you will find that it has more saturated fat. And it
Oh,
would
okay, I
have
thought
less.
this is just like what, this is like preferred, what you want to happen, but.
Oh,
not what
yeah, no.
happens.
So that's actually what happens.
Oh, okay.
Okay. Do you get this part
That's
now?
yes.
Now, so how do listen, right? How do you achieve a more solid cell membrane? You need to have more saturated fat because we know saturated fat packed together closely, right? Because the tails are straight, right? Remember I said it's like people lining up with the hands down, right? So if they're able to pack together closely, then they will create a more solid form. Okay? Remember, saturated fat are solid at room temperature, right? So you want more of that and then less of the other two. All right. Hopefully that's a little bit better. All right, next. We are going to look at 6 membrane protein functions. Okay. I would memorize maybe 4 of them for the test. Okay. Just be able to name them and understand what it is. I'll give you a full review sheet for the test later on, right? But I just want to tell you ahead of time, okay? In case you can start early with your study. So all those membrane proteins, like I said, some can go through, which would be integral protein. Others are just on the side, which would make them peripheral proteins. Okay. And they all have important functions. And we are going to look at 6 of these functions, starting with the first one. So here, these are red blood cells that you're looking at. And red blood cells are has a very distinct shape. You can see there's a little indentation in the middle, okay? So how do you maintain that indentation, okay? So, bless you, when the cell membrane caved in, right, basically, you need to have some kind of membrane protein to anchor the membrane in place, okay? So like this, think of the cell membrane as like a sheet, all right? So what you need to do is you actually need to hold in a little bit at the middle, okay? You pulled in a little bit at the middle of the sheet, okay? To create this kind of indentation, right? But something has to hold on to it, right? What's going to hold on to it? Well, that's going to be like some kind of membrane protein. It's like setting up a tent. Okay? You need to anchor the tent to the ground so it's not going to get blown away, right? And that membrane protein is the anchor. Yeah,
Can I have a question?
sure.
What about the nucleus of cell?
Yeah, for this one, right? Yeah, excellent question. Because you're thinking the cell membrane is like caved in, there's no space for this nucleus, right? And you're absolutely right. Red blood cells are one of the few cells in the body that do not have a nucleus. Okay, so even though they're eukaryotic cells, they don't have a nucleus. Okay. So that's number one, attachment to cytoskeleton. Number two, cell signaling. Your body communicates with different parts using a lot of signaling molecules. Some of these are hormones, others are just chemicals, okay? And even though you release these chemical signals throughout the whole body and all of your cells are exposed to them, they do not always respond to them. In order for the cell to respond to the chemical signal, they have to have the right receptor for it. Those receptors are membrane proteins. For example, if we release these molecules that have a little triangle shape at the end, the cell membrane protein needs to have the correct fit for it. Okay? If the membrane protein looks like this with like this kind of groove, then the little triangle is not going to fit and it's not going to respond to it. Okay? So that's what the cell signaling does. Now, I'll give you an example. Adrenaline, or some people call it epinephrine, is the same thing, right? Like an EpiPen, epinephrine. Adrenaline is released from your adrenal gland, which sits on top of your kidneys. Can you show me where your kidneys are, roughly? It's at the front, at the back, at the side, the top, where? It's over here, okay? So that's where your kidneys are. And on top of the kidneys, you have the adrenal gland. Yeah, it's not down here. That's the butt cheek, right? And so, anyways, on top of the kidney, you would have your adrenal gland, which secretes adrenaline. Okay, and you would secrete adrenaline when you are excited, when you are in danger, right? Okay, you guys might have heard of the fight or flight response, right? And when like there's something dangerous is moving towards you, you either run away or you have to fight it, right? So that's adrenaline. Right, but a person getting ready for race, right? They could pump themselves up and get their adrenaline going as well. Okay. Now listen, what does adrenaline do? Adrenaline, once it's released, it's gonna go through the entire body, but only cells with the right receptor will respond to it. Okay. So in this case, this is a cell and it has the right receptor. The adrenaline will bind to it, and it will cause a series of reactions to change the behavior of the cell. In this case, the person is trying to get ready for a race, so it makes sense for the cell to do hydrolysis of glycogen. You remember glycogen is storage form of energy, right? So you're going to release those energy in the form of glucose so that the person has the resources to run. Same thing with the fight or flight. If you're in danger and you need to run away or fight the bear or something, you need those energy, right? So that's what the receptor does. It takes the signaling molecule and then it changes the behavior of the cell. The signaling molecule never went into the cell, right? It just binds to the receptor and the receptor relays the information. Yes.
So when someone faints,
Sure.
for example, what would cause that? Would that be glycogen or like you don't have enough?
Well, I mean, there are various reasons, right? But it's possible that you don't have enough sugar going up to the brain, right? Then, yeah, or your blood pressure is too low. Those are reasons that might lead to the fainting. So let me tell you a story to help you remember this, okay? My family, they are anti-Halloween, okay? Growing up, every year when it's Halloween, my mom doesn't like the concept of handing out free candies, okay? So what happens is whenever someone rings the doorbell to get candy, my mom will be like, turn off all the lights, hide in the basement, okay? Pretend no one's home. And I also always think, Mom, they can see you turning off the light, right?
Mhm.
Like something is suspicious when they ring the doorbell and then the light goes, they know. Okay, but you know, I don't talk back to my mother. Anyways, so it's like this situation here. You see, the kids who are ringing doorbell, they never came into our house, but it causes all these commotions, all these changes and activity inside the house, right? And that's what the membrane protein does. It's like the doorbell. It relays the information from outside and it elicits a bunch of change in behavior inside the cell. Okay? So without the receptor, then the signaling molecule is not going to achieve anything. Do you understand? You actually do this in the brain as well, but we'll learn that next semester. Okay, I think we're gonna leave it at that for the moment, and we're gonna have a break. Please sign yourself up with the thing over there for the lab. Okay, I also created this slide. I'm just gonna let it run. Okay, so we were talking about the functions of membrane proteins. Number one, it is to attach to cytoskeleton. Number two, it is to form a receptor protein to help relay signals. So we're gonna have 4 more to come. Number 3, enzymatic activity. Right? We already know enzymes are proteins and they help speed up chemical reactions by lowering the energy requirement. Okay? But sometimes we need to do multiple reactions one after the other. And in those cases, it is actually easier to put the related enzymes right next to each other. Right? So enzyme 1, 2, 3, 4 are all related to the same pathway. Right? So when the ingredient goes into one, it spits out a product, and immediately it could do the second reaction, and so on and so forth. So by putting these enzymes close to each other on the membrane, it allows the reactions to happen quickly and efficiently. It's the same reason why we put all our cooking stuff in the kitchen. You're not gonna leave your knife in the bathroom and then the cooking pan in the garage or something, right? Like then you will have to run around and get them when you need them. You put them all in the same place so you can grab them easily and quickly when you need to use it. That's why we put all the enzymes together on the membrane rather than having them randomly scattered in the cytoplasm. All right? So that's function number 3. Number 4 is something that we've talked about previously. Oh, no, it was another class. So number 4 is an intercellular junction. In various locations of our body, we need to create tight connection between cells. We don't want blood to be leaking out from our blood vessels. We don't want food to be leaking out of our intestinal walls. So those places, we need to staple the cells together pretty much. And that's what the membrane protein allows us to do. They are basically glue and staplers that would seal adjacent cells together. So things are not going to leak between them. Right? It prevents uncontrolled movement of materials between cells. All right. Number 5, cell recognition. The sugary protein or the glycoproteins and the glycolipids create a sort of ID tag on the cell. And your immune system is familiar with that ID tag. If it detects something in the body that does not have the same ID tags, they will attack it and destroy it. Okay. The underlying principle of how your immune system works is the ability to distinguish self and non-self, right? To tell the difference of what belongs and what does not belong to the body. If your immune system fails to do that, they might, number 1, end up attacking your own cells, and we have what's called autoimmune diseases. Do you guys know any autoimmune diseases? Diseases where your body turns on yourself and attack it? Yes?
HIV.
Not quite. Yeah, that would be immunodeficiency, which one? Yeah. Yeah.
Spot,
Pot?
like Pott's disease.
Or POTS?
POTS,
Or POTS?
POTS.
I don't know what that is. Sorry. I'll look it up. Yeah. Anyone else? Lupus, yes, lupus is autoimmune disease, right? You might have heard of multiple sclerosis, right? That's another autoimmune disease. Anyways, we'll look more.
Static tachycardia.
Okay, I know tachycardia, but I don't know the tachycardia is like your heart rate going too up.
Goodness.
I will look that up after class. Okay. Anyway, so that's a cell recognition. You guys okay? Here we have a white cell, and here we have a cell that does not belong to the body, and it's gonna attack it.
It's a nervous system disorder.
Number 6, transport. That's the main focus of lecture 4. Okay, transport, to move things across the cell membrane. Some molecules, as you will see, is able to cross the cell membrane on their own, but others require a little bit of help. And usually the help comes in the form of some kind of membrane protein. Okay, so we will do a big note on that later on. Okay, so remember, there are 6 functions associated with membrane proteins, and you should memorize at least how many of them? 4, okay? Just memorize 6 if you could. Excellent. Okay, but if you're running low on memory space, 4 is okay. All right, let's see how much you remember. We are looking for the true statement here. It's going to take some time to read. Good.
Which of the following is a true statement?
I would. Good try. Try again. Try again, please. I didn't see that, May. What was it? Which one is it? No, try again. Good. Anyone else? I can't see Kim. Which way? Yes, very good. Any last try? Yes. Good try. Not quite. Is it an answer, Amir, or you just think this that you're thinking, look? No. I see some fingers here. Yes, very good. Okay, here we go. We're looking for the true one. First one. They as in the phospholipids, they are free to depart from membrane and dissolve the surrounding solution. That's not true, right? If that's the case, then the cell membrane would just dissolve and fall apart on its own, right? Killing the cell. They occur in an uninterrupted bilayer with membrane proteins restricted to the surface. They don't have to be on the surface, right? They could be on the inside, or they could be going through the membrane. Okay, so that's why this is not correct. They have hydrophilic tails in the interior of the membrane. The tails are hydrophobic, right? They can move laterally along the plane of the membrane. That's the correct one. Remember, I told you the balls can like bounce around, right? But they do not flip, right? They do not flip. So that's why this is not correct. No fifth fall. Okay? So that's the answer, D. So last quiz, some of these questions were on, right? I don't know if you recognize it. Okay? If you don't, they are, okay? So this time, make sure you study these as well. I'm not saying this one is on, but like, some of these might be. Well, this is not even going to be on your quiz anyways, right? This is a different lecture, right? But like I'm talking about lecture 2 and 3. Okay, how about this? This one should be straightforward. Yes. Yeah. Good. Anyone else? Good. Angelica, Monique, yeah. Anyone else? Yes, Annie? Yes. Anita. Anyone else? Try again. Okay, it's the 6 things that I asked you to remember, right? So you just have to find the one that's not Kim, that's correct, yeah. So protection from lysis, that's That's not membrane function, right? That would be the function of what? We learned it earlier today. What prevents the cell from bursting?
The contract, don't.
No. Oh, okay. Yeah, okay, yeah, true. But that's not what I'm thinking though. Yeah, go ahead. Oh, you're gonna say that? Okay. Sure, yeah, the contractile vacuoles will pump out water and prevent them from bursting. Sure. I was also thinking about cell wall. Okay, yeah. But yeah, I guess both would be correct. Okay. Anyway, the answer is D, all right? D. I will give you marks for both. Yeah. Okay. Okay, we're gonna talk about cellular energy for a little bit. All the chemical reactions that's happening in the body, in the cell, we collectively refer to them as metabolism. Okay, metabolism. But it turns out there are two kinds of metabolic reactions, depending on whether you're breaking things or whether you are building things. If you are breaking things, then they are called catabolic reaction or catabolism. If you're building things, then it's called anabolism or anabolic reaction. Let's write this down together. Now, if you have the notes print out, there is a diagram like that. If you don't have it, that's okay. Just, it's easy to draw. Okay, so we'll fill it out together. So over here, we start with macromolecules in the food that we eat. I'm sure, or at least I hope you know what they are by now. Carbohydrates, right? What else? Just yell them out. Yeah. And lipids, right? So we eat them, consume them, goes into our body. And then we need to break it down. I'll make it bigger for people at the back. So when you break these things down, we call it catabolism. Catabolism. Okay, or catabolic reaction, same thing. Catabolism. Break things down. The way to remember it is the prefix kata, like catastrophe. In a catastrophe, earthquake, tornado, everything breaks down, right? So in catabolism, we are breaking molecules down. When we break the molecules down, we will get some energy from them. And then we can use that energy for something else later. But we are not very good at it. We are not very good at converting food into energy. Much of the food that we metabolize is actually going to be converted into heat instead of energy. So here's some background information for you. If you think about like a traditional light bulb, the really old school ones, they are about 10% efficient. Okay, what that means is 90% of the energy is wasted as heat. Only 10% is converted into light. The really old one, with the filament on the inside, right? That's these incandescent light bulb, right? So they're not very efficient, right? Waste a lot of energy. And then we have the newer ones, the one that looks like the spiral. Right? Those are called fluorescent light bulbs, right? So those are a little bit better. They are about 80, actually a lot better, 80% efficient, right? So they're not very hot. Even after you use it for some time, you touch it, it's okay. It's tolerable, right? They only lose about 20% of the energy. And then what's the really energy saving one? The new ones. What do we call them? There are three letters. That's right. LED. LED are 90% efficient. Okay. They only waste 10% of the energy. You touch it, you don't even feel any. So what about our body? Do you think we are like the incandescent light bulb, fluorescent light bulb, or LED? I kind of told you we're not very good, but like give me a number. How efficient are we? Wild guess doesn't have to be.
Or beat?
Yeah, us, yeah, our body. Give me some numbers. 13%, that's very precise. Yeah, anyone else? You guys are no fun. It's okay to guess.
I say.
Our body. Our body. There we go. 25%, okay? We are about 25% efficient. And we can calculate it because we know the kind of reactions that are going on. And then, we can do calculations to figure out how much of it is wasted. So we are wasting 75% of the food's energy as heat. But it's not too bad, I guess, because we are warm-blooded. We need to maintain a core body temperature of 37 degrees, right? So all that heat is going to help us stay warm, basically. Right? So especially on a cold day, after a meal, you feel warm. It's because your body's starting to break down these things and you generate heat. Okay? So what are you breaking the food down into? Well, you're going to break it down into various building blocks. Okay? You might have, I'm just going to put a few examples here. I'm sure you already know amino acids, right? Okay, you might have some fatty acids, all the building blocks that we learn. And then your body, your cells will use these building blocks to build molecules that you need in the body. Okay, so these are macromolecules needed in our body. And to build them, we call it anabolism. Okay, so instead of breaking things down, we are building things. And building things takes effort. It takes energy. And that's where we're going to use the energy. Essentially, the energy produced by catabolism will be used to enable anabolism. If we don't eat, we don't get energy, we can't build anything. Okay. So let's say you want to build insulin, which is a protein, right? You can't just eat insulin and use it. It doesn't work like that. You eat proteins from meat, from soy product, then you get amino acid from it, then you build the insulin. Yes?
Can you repeat one more time? Energy produced by catabolism?
Is used to drive anabolism.
Thank you.
Yep. Is that okay, everyone? Yeah,
Go back.
sure.
Because. So what is this, these like things at the bottom?
Yeah, so these are the building blocks, right? Remember proteins are made of amino acids, so now you get the individual amino acids,
Oh,
which then
okay.
you can build. Yeah, it's
Oh.
like a Lego set, right? You break it down and then you build whatever you want to build with it. One of the key catabolic reactions in our body that provides energy for us is cellular respiration. We saw this a couple times already, right? In fact, at the beginning of today, I told you cellular respiration involves taking glucose, reacting it with oxygen to produce carbon dioxide, water, and energy. Part of this reaction happens in the mitochondria, and we will learn about it in lecture 5. Okay, lecture 5 is the hardest lesson of the whole unit, of the whole first half of the course, which is why we leave it till the last. Till the end. Okay. A very important molecule in the body is called ATP. So let me write it down for you here. ATP is the currency of energy in a cell. Okay? You can think of it as a battery. ATP stands for adenosine triphosphate. So it has the adenine. It's the same adenine as DNA. It has the ribose sugar, and it has 3-phosphate. By now, you should know phosphate is PO4, right, from chem, and there are 3 of them. That's why it's tri. So in this form, in the ATP form, this is fully charged. Fully charged. And you can use the energy from ATP by snapping off one of the phosphate in the end. So if you break the phosphate off, then now it becomes ADP, adenosine diphosphate. Adenosine diphosphate, only 2 phosphate. And this is, I guess you can say it's drained. Okay. So it's empty now. If you want to charge it up again, then you have to provide energy, and then you put the P back on, and it's ready to use it. So at any given time, your body would have a pool of ATP charged batteries at your disposal. You can use it for different reasons. Once you use it, you have a bunch of empty batteries, ADP. But we don't throw them away, right? They're rechargeable, just like your phone is. Just give it energy and it will go back. So it's cycling between ATP and ADP. Do you understand? Okay, let me put it into perspective for you in relation to the catabolism and the anabolism. Okay, so a new diagram. So let's say we have macromolecules, and then we have building blocks. This is the last thing, okay? And then I'm gonna let you go. If you break down macromolecules into building blocks, which one is it? Is it catabolism or anabolism? Yeah, very good. And then it releases energy, right? And heat, yeah.
Okay.
But we don't want to waste these energy, so we have to store it. And that's when you're going to have the ADP, the empty batteries, and you need to find the cap for it, the P. And these things will trap the energy, and then it becomes ATP. So that's the breaking down the fluid supplies the energy to recharge your battery, recharge your ATPs.
Wait, can you repeat that?
Sure.
Wait, do I have to write building blocks or can I just write amino acid?
Well, it's not always amino acid, right? If you're breaking down carbohydrates, then it becomes glucose, right? Yeah, so building block would be better. So catabolism provides energy to be stored by ATP. Remember, the advantage of having ATP is you can trap the energy and use it later. Okay? It's not like you have to use the energy as soon as you produce it. That would be like solar energy, right? Like you have to use it when the sun is out, right? But if you can store it, then you can use it at a later time. That's what the ATP is for. Okay? You can store these energy until you need it. So what do we need it for? Well, one example of where we need it is anabolism, right? For building things, building macromolecules in the body. And so we take the battery, we kind of crack it open, so to speak, and we're going to change it back to the drained form. But more importantly, in this process, you're going to release energy. And that allows you to do the reaction to build things. It does? Yeah. Good. I lied. One more slide. I just remember we have one more slide. Sorry. I'll post this, okay, if you haven't finished copying it. What else do you use ATP for? So I'll give you 3 more examples, and that's it. You need ATP to do mechanical work. Mechanical work means moving things. Okay, so we move a lot of things in the body. Yeah? You move your muscles, your heartbeats, your breathing muscles, right? All those movements requires ATP. Okay, so here we have a cell with 2 flagella, and the beating of those tails needs ATP. Doing chemical reactions need ATP in some cases. Okay, like the anabolic reaction we talked about, that requires ATP. And finally, the focus of next class is going to be transport work. Whenever we try to move things from a low concentration area to a high concentration area, that's going to require energy in the form of ATP. From high to low, it's okay. It happens on its own. But from low to high, it requires energy. For example, if I have an object here, I let go, it automatically goes from a high point to a low point. But picking it up, that requires energy. Okay, think of this as like a 20-pound dumbbell. To pick it up, that requires energy. Going from low to high is going to need ATP.
So why do some people have like more energy than others?
Yeah, it depends on your metabolic rate, right? So some people, they burn up their fuel faster,
Yeah.
and then they feel more tired, right? Yeah, that's one reason anyways.
Or is it just that you're not like you know how like, okay, someone that like runs all the time?
Yeah,
They
it could
go
be
with
too.
that.
Your body can get adapted to it, right? Yeah, exactly, right? The more you train. Okay, that's it, guys. Thank you. Study hard. Hey, quiz on Tuesday. Lesson afterwards. Drink lots of caffeine. See you then.